Hello students, welcome back. In the last lecture we have covered the applications of biotechnology in agriculture till somatic hybridization. Today we will be discussing few more applications of biotechnology in so you can see the one important application is genetically modified organisms. What are genetically modified organisms? There are three words in this. In short, you call it as GMO. Genetically means you are doing modification or changes in the gene or genome of any organism. Modifying the genome of any organism. So this is possible in various type of organism, plants, bacteria, fungi, animals. In plants we do these modification generally in the domesticated plants for following reasons, like making the plant more tolerant to abiotic stresses. Reduction in the application of fertilizer means we are improving the yield potential of the crop, so that we need lesser amount of nutrients from the outside. Also it is also useful in increasing the resistance towards the pest. Some problems are faced with the harvesting of crop is lodging, majorly in the cereal crops. So to prevent the lodging and other kind of post harvest losses, genetic modification helps in it also. Then it also increases the nutrient quality, or you can say nutritional composition of the crop. You have heard of biofortification also, means you are improving the quantity or amount of any particular type of micronutrient or a mineral in the crop by modifying the genome of the plant. One of the example of this is golden rice. You have to remember the examples, because in both or in other examples are very important. Examples are directly asked. The most important example used in genetic modification is Bt cotton. Bt cotton is a cotton plant which is modified against a pest. So it is made a pest resistant plant. For which pest it is modified? It is modified for the pink bollworm. It is modified against pink bollworm. It is a type of pest which attacks on the balls, cotton balls, which are at the bud stage. It attack on the cotton ball and it destroy it, so that reduces the yield of the cotton. So what happens in this Bt cotton? This Bt cotton, the cotton plant, the gene of the cotton plant is inserted with a gene, that is cry gene, from which organism? Bacteria, Bacillus thuringiensis. The Bt word comes from Bacillus thuringiensis. This Bacillus thuringiensis gives a gene which is toxic to this pink bollworm, and that gene is cry gene. It produces cry protein which perforates the stomach, which perforates the gut of the this pink bollworm. When this cry protein is insecticidal protein, you have to remember the protein. The organism is very important. Bacillus thuringiensis is the organism from which this gene is taken, and it is incorporated in the gene of the cotton plant. What it does? It produces a toxin which is inactive in plant, but when it enters in the gut of the your pink bollworm, then it gets activated due to the alkaline pH of the alkaline pH of the gut and start forming the toxic chemical compound and does the perforation, creates the pore and causes swelling and death of the insect. Okay, this specific Bt cotton genes are isolated from that I already told you. You have to remember the names of the toxin which are coded by gene CRY-AC1 named as CRY. Okay, so these are important genes, CRY1AC and CRY2AB, which are help in controlling cotton bollworm, and CRY1AC is helpful in controlling the bollworm insect, and CRY1AB is helpful in controlling the corn borer. Okay, so these two different genes are helpful in controlling different type of insect. Okay, now another example of pest resistance through genetically modified organism is nematodes. Nematodes are the another type of pest which attacks on the root. Nematode basically attacks on the root of the plant, and this is done in the tobacco plant. Okay, the the Bacillus thuringiensis is used in cotton, and this nematode resistance plant is produced in the tobacco. There is a nematode which is named as Melidogyne incognitia. Melidogyne incognitia, which is basically affecting the roots of the tobacco plant and reduces the yield. Of course, if the base of any plant is get damaged, so it won't be able to get nutrients and get flourished. Therefore, roots are very important. So to prevent the root death, this Melidogyne incognitia nematode has to be controlled. There are various chemicals used, but still it can't be controlled. If the infection is there in the roots of tobacco, then that plant has to be removed. So to prevent that, what is does, a strategy has been adopted which is known as RNA interference. RNA interference generally takes place in eukaryotic organisms as a cellular defense. It is a defense mechanism already found in the eukaryotic organism. What happens in this? This method involves silencing of specific mRNA due to complementary double RNA molecule that binds to and prevent translation of the mRNA. Basically, what happens in this technique, RNA interference, RNAs are formed in every kind of organism. So whatever RNA is formed by the nematode, you can I think no picture is there, so you can see here, whatever RNA is formed by the nematode, that RNA is harmful for the plant. So what it happens, the gene which is inserted in the plant, what it does, the RNA formed by the nematode, it is get coded by the gene RNA produced by the plant into a double-stranded RNA. Once the RNA becomes a double-stranded RNA, it will not does any kind of problem in the plant. So it is also known as silencing, RNA silencing. What happens? Due to complementary double-stranded RNA molecule which binds to the RNA produced by the nematode and silence the process of translation, means this RNA will not produce protein, and if protein is not produced, then root will not be damaged. So basically the RNA produced by the nematodes are silenced with the RNA produced by the genes inserted in the plant, which prevents the protein formation which are toxic to plant. Now, which RNA, which gene is used, or from where we use the gene, is agrobacterium. Agrobacterium is the vector which is used to insert the nematode specific gene. For every kind of nematode, you need to have a different kind of gene to silence its RNA. It's not like that. For melidogyny in cognitia, you are using a specific type of gene to silence its RNA. Same type of gene is used to silence other nematodes RNA. No, you have to use a different type of gene. Specific gene is there for a specific type of nematode, so you have to use that one. With the help of agrobacterium vector, you will be inserting the specific gene in the plant, and that will make a complementary RNA strand and silence the RNA of the nematode. That is RNA interference. Okay, you can see in the image where in the part A, this image shows that this root is affected. B is root of a transgenic plant, means A is showing the nematode affected root, and the picture B is showing root which are not infected because the RNA is silent, so root is not infected by the nematode. Okay, transgenic plant roots 5 days after deliberate injection of nematode but protected through novel mechanism. So in this, to check that nematode is affecting, deliberately nematodes are inserted in this region, rhizosphere, where it is found that there is no infection after deliberately infecting the plant with the nematode. Okay, so these are the different applications of biotechnology in agriculture. We can quickly see the applications once again. Tissue culture is one of the application. Another is micropropagation. Then you did the somatic hybrids, you form the same kind of hybrids, and then finally you use genetically modified organism for increasing, for various purpose, for increasing the yield, for making pest resistant, for increasing the nutritional value of the crop or plant, and also for making the plant tolerant to various type of stresses, whether it is biotic stress, whether it is abiotic stresses. So these are the application of biotechnology in agriculture. In the next topic, we will be seeing the application of biotechnology in medicine. Thank you for watching.